Nature is a master engineer. It has perfected astounding feats of biological complexity. One such marvel is regeneration, the ability to regrow lost or damaged body parts. This phenomenon is observed in various species across the animal kingdom. Starfish can regenerate entire limbs from a single arm. Planarian flatworms can regenerate into multiple individual organisms. Salamanders can regenerate complex structures like limbs and tails throughout their lifetime. For example, if they lose a limb, this limb will grow back. Salamanders can grow back their eyes, their jaws, their hearts, portions of their brain and ovaries. And throughout adulthood, they restore perfect copies of the original. So we know regeneration is possible if we learn how to control the anatomical structures that cells build. Understanding these mechanisms could revolutionize medicine. By deciphering the secrets of regeneration, we can potentially heal and regenerate our own bodies. At the heart of regeneration lie stem cells, the body's own master builders. These remarkable cells have the unique ability to differentiate into various cell types. They are essential for growth, development, and repair. Stem cells are like blank slates, capable of becoming any type of cell in the body. Another crucial aspect of regeneration is cellular communication. The intricate language cells use to coordinate their actions. Cells communicate through a complex network of signaling pathways. These pathways involve molecules that act as messengers, relaying instructions and information between cells. In the early stages of embryonic development, cells communicate and cooperate to form complex tissues and organs. This process is guided by our DNA, the blueprint of life. Understanding how cells interpret and execute these instructions is crucial for unlocking the secrets of regeneration. Imagine a world where we can manipulate these processes. By directing stem cells and controlling their communication, we could potentially regenerate damaged tissues and organs. This could revolutionize the treatment of injuries and diseases, offering a new era of personalized medicine. Regenerative medicine holds immense promise for treating a wide range of injuries and diseases. Spinal cord injuries, which often result in permanent paralysis, could be potentially reversed by regenerating damaged nerve cells and restoring connections. Cancer, a leading cause of death worldwide, could be treated by selectively targeting and destroying cancerous cells while promoting the growth of healthy tissue. Heart disease, another major health concern, could be addressed by regenerating damaged heart muscle, improving cardiac function. Diabetes, a chronic metabolic disorder, could be managed by regenerating insulin-producing cells in the pancreas. The goal of regenerative medicine is to convince cells at the injury site to rebuild the damaged structures. This is akin to providing the body with the necessary tools and instructions to heal itself. Imagine a future where organ transplantation is no longer necessary because we can simply regrow damaged organs. This is the ultimate goal of regenerative medicine, to harness the body's innate healing potential to restore health and extend lifespan. Pioneering the future advancements in regenerative technologies. Scientists are making significant strides in regenerative medicine. Tissue engineering focuses on creating functional tissues and organs. Using biocompatible materials, they create scaffolds that support cell growth. Stem cell therapy transplants stem cells to promote regeneration. Researchers explore embryonic, adult, and induced pluripotent stem cells. Three-dimensional printing is revolutionizing regenerative medicine. It allows the creation of complex structures like scaffolds and organs. The science behind regenerative medicine is advancing fast. One researcher that is unraveling the complex mechanisms that allow organisms to regenerate is Dr. Michael Levin, director of the Allen Discovery Center at Tufts University. His research focuses on how electrical signals control cell behavior. Let's listen to him explain important aspects of his research.
Here at the Allen Discovery Center at Tufts University, we are learning to understand collective decision-making by cells, and we're finding ways to control biological growth and form to develop new biomedical treatments. Now, I want to introduce you to a kind of animal called planaria, and these are really the champions of regeneration. They can be cut into many pieces, in fact hundreds of pieces, and every piece will regrow exactly what's missing, no more, no less, to give a tiny, perfect little worm. In fact, these animals are so regenerative, they've achieved immortality. There's no such thing as an old planarian, and every piece will continue to regenerate perfect, healthy new tissue. They are, in fact, so regenerative that they are teaching us something very important about the relationship between the mind and the body. It was found that planaria that are taught a particular fact, such as that food can be found in a certain spot in their environment, can then recall this fact even after their heads are amputated and grow back. So the information that a planarian picks up during its lifetime is stored throughout the body, and this information is imprinted on a brand new brain when the head regenerates. This is telling us that there's a profound relationship between the information that is learned throughout the lifetime of a creature and the body structure that forms its various organs, including its brain. What really is a memory? How is it stored, encoded, decoded, and propagated from various body tissues into the brain and vice versa? This addresses not only profound topics in the philosophy of mind, but also the biomedicine of degenerative brain disease, because at some point, human patients will have cellular therapies delivered into their brain, and we need to understand what will happen to the personality and the memory of such patients. So these model systems are helping us to understand some very basic questions about memory, personal identity, and the controls of organ structure. Biology today is focused on the molecular hardware. Many labs are getting very good at manipulating individual genes, proteins, and pathways. But what we need to do in addition to this is learn to control the software of life. Several key insights allowed computer science to drive the remarkable revolution in information technology that we see today. Back in the 40s and 50s, the way to program a computer was through physical rewiring. And you can see she's doing this right here. In order to get this machine to do something new, she would have to change the wiring. She would literally have to physically alter the structure of the machine. But what computer scientists figured out is that if the hardware is good enough, then you can control it with software, meaning you can control it with stimuli or inputs that change its behavior without having to change the structure of the machine. And this gave rise to lots of applications that we use every day that show us how powerful this concept of reprogrammable software really is. And so I want to show you a new path that we found to be able to do the same thing in biomedicine. Now, in the brain, we have some hardware which consists of cells known as neurons, and these neurons communicate with each other through channels known as electrical synapses, and they conduct electrical impulses. And this network of electrical impulses supports a kind of software. So here you can see the electrical activity of a zebrafish brain while this zebrafish is thinking about whatever it is that fish normally think about. And it's possible now to begin to decode the information content or the thoughts that are driven by all of these sets of electrical activity. Now, what we found out is that in fact all cells, not just neurons, have this ability. All cells drive electrical signals. All cells communicate with each other through various kinds of electrical connections, and it turns out that this ability to store and process information in electrical networks is universal. And what you see here in the bottom right is an early frog embryo, and all the different colors are showing you in time-lapse video all the electrical conversations that cells are having with each other to determine which cells will make the right, left, top, bottom, anterior, and posterior parts of the developing tadpole. And these bioelectrical patterns are an extremely important instructive influence over what happens in embryogenesis. So these patterns are what we call bioelectric scaffolds or bioelectric pre-patterns. Here you see another time lapse of a frog embryo putting its face together. And so here's one frame from that video, and you can see the white regions are cells which have a very particular bioelectrical property. They're depolarized, and you can already see in this new tissue, you can already see the outlines of the frog face. There's the eye, the mouth, the organs on the sides called placodes. You see all of this that's going to be formed later on. So this is an instructive pre-pattern, and if this pre-pattern is disrupted, then the gene expression will change, and then the anatomy will change. And in fact, we have examples of pathological changes. Down here you see a tadpole that bears a human oncogene, which is going to induce a tumor. But even before that tumor becomes obvious, you can detect it non-invasively using a fluorescent voltage-sensitive dye, which reveals that these cells are going to have an aberrant bioelectrical potential, and they are not going to cooperate with the rest of the body. They're in effect shutting off the electrical connections with the rest of the tissue and going off on their own. They're going to treat the rest of the animal as just environment, and they're going to revert to a kind of unicellular ancient existence, and this is what we see in metastasis and cancer progression. So watching these bioelectrical signals is really important, not only for understanding where developmental anatomy comes from, but also as potential diagnostics for birth defects and also for cancer. Now, what we've developed are ways to control these electrical properties in cells using pharmacology and light. So we use drugs to open and close these ion channels that are proteins that produce electrical signaling in every cell, and we can use these drugs together with light stimulation to provide various kinds of electrical pre-patterns directly to growing tissues. And so one application of this here is in the normalization or reprogramming of tumors. Here you see a tadpole embryo that has been injected with an oncogene, and normally it forms a tumor, and you can see that here with these labeled in red. But even though that oncogene is strongly present, you can suppress the appearance of the tumor by co-injecting with the oncogene a specific RNA encoding an ion channel that normalizes the electrical properties of those cells. So by controlling the electrical properties, even though the genetic mutation is present, you can suppress the appearance of the tumor. You can force those cells to cooperate with their neighbors to remain in electrical connection, and by enforcing the correct bioelectrical state, you get normal tissue instead of a tumor. We've also used the same technology to repair brain structure in tadpoles that was damaged by mutations or harmful teratogenic drugs. So here is what a normal tadpole brain looks like. A truncated, misshapen brain results from mutations in genes like Notch or in exposure to alcohol, nicotine, things like that. And what we found is that by manipulating the native ion channels in a very particular way using drugs, in fact drugs that are already approved for human use, we can restore the bioelectric pattern that is appropriate for telling these tissues how big and what shape a brain they need to have. And in fact, then the correct brain is built. And these tadpoles have not only the normal shape of brain, but they have normal gene expression, and they even have normal IQs. So we now have a computational system, a set of programs that allow us to find the correct drugs to induce an appropriate bioelectric state to repair some very drastic craniofacial defects. And in fact, what we also discovered is that tissues have anatomical memory. So here's a planarian, and normally when you cut a planarian into pieces, they give rise, every piece gives rise to a normal flatworm, one head, one tail. Now we can perturb briefly this bioelectric circuit after cutting and confuse the piece about how many heads it's supposed to have. In fact, we can reset that bioelectric pattern to dictate having 2 heads. And in fact, the middle piece here has grown, you can see that here, has grown 2 heads. But the most amazing thing is that if you then again take the middle piece out of that worm and culture it again in plain water with no more manipulation of any kind and simply let it regenerate, it will once again make a 2-headed animal, despite the fact that it has a normal genome, a normal genomic sequence that we have not edited. And so in perpetuity, as far as we can tell forever, every time you cut these worms, you get a 2-headed animal. So think about what this means. The pattern to which these cells build after damage can be rewritten, can be bioelectrically respecified towards 2 heads, and in fact now we can set it back to normal as well, in a way that only changes the information stored in this electrical circuit. The DNA is completely normal. We have not changed the genomic sequence of this animal. So we have now found that some information is present in the physiological circuit that is not directly in the genome, and this physiological circuit can store information that is changeable by us. In fact, not only can you ask these worms to make a second head, if you perturb the bioelectric network, you can also get them to build heads that are appropriate to other species of worms. So here you see a normal Dugisia doratocephala with a nice triangular head. By disrupting that bioelectrical gradient, you can make their fragments regenerate flat heads like a Piphalina or round heads like an S. mediterranea. And not only the external shape of the head changes, but also here in green that you can see the shape of the brain becomes like these other species, and the distribution of their stem cells, which you can see with the red dots, are also changed to be just like these other species. And again, this is done without changing the genomic sequence. So extremely important information is present in this bioelectrical network that guides cell activity towards building specific different kinds of structures. A very important thing to understand is the concept of nested cognition. At all levels of organization in biology, from molecular networks to subcellular structures to neural networks to whole tissues, organs, whole organisms, and even groups like ant colonies, there is the ability to solve problems at every scale. Every one of these levels of organization has a degree of intelligence that allows it to solve various challenges and changes in their environments and maintain optimal function. If we understand the scaling of this ability, how it is that the activity of the simpler levels underneath give rise to intelligent activity at higher levels, we will achieve transformative applications in medicine and artificial intelligence. The idea is not simply to understand the mechanisms by which these things happen, but to understand the information, how every layer of the system is able to produce more complex, intelligent responses and robustness at higher levels. This will not only allow us to have much better control over organogenesis for regenerative medicine, but also we can use this kind of information to develop new ways of achieving artificial intelligence. In summary, all cells communicate electrically to form networks that make decisions about growth and form. By reprogramming this physiological software, we can unlock the promises of regenerative medicine. We can go beyond genomic editing of the hardware of life to provide definitive solutions to injury, aging, and cancer by learning to convince cells to build whatever we want them to build. I want to thank the postdocs, collaborators, and students that have contributed to the work that I've shown you today. And of course, I want to thank my funders who support our research. And I thank you for listening.
As we can see from Dr. Levin's explanation, these technologies are paving the way for regenerative medicine in healthcare. A vision of tomorrow: regenerative medicine transforming healthcare. Imagine a future where regenerative medicine is an integral part of healthcare. Hospitals are equipped with state-of-the-art facilities for tissue engineering, stem cell therapy, and three-dimensional bioprinting. Personalized treatments tailored to an individual's genetic makeup and specific medical needs are commonplace. Injuries that were once considered permanently debilitating are treated with regenerative therapies, restoring mobility and function. Chronic diseases like diabetes and heart disease are managed effectively, improving quality of life and extending lifespan. The ethical implications of these advancements are carefully considered, ensuring responsible and equitable access to these transformative technologies. This vision of the future is within reach. Continued research and investment in regenerative medicine are crucial to unlocking its full potential. By supporting scientific exploration and fostering innovation, we can make this vision a reality. The promise of regenerative medicine is vast, offering hope for a healthier and more vibrant future for all.